cell, we see a nucleus, and inside the nucleus, when we enlarge the nucleus, we see these structures which are chromosomes. And when we take this section of the chromosome, we see this double helical structure, which is DNA. Out of this DNA, 98% is junk and has no role to play, but 2% of that DNA makes protein. And that segment of DNA that codes for a protein is a gene. Okay, so I hope the difference between these terms is clear to you, and you should be able to define them in the exam. Now, when we talk about the DNA structure, it is composed of a phosphate, sugar, and the phosphate present, the sugar present is deoxyribose. That is why DNA is known as deoxyribonucleic acid. And this phosphate and sugar, you can see it forms a backbone of the double helix. And within them, there are nitrogenous bases, and there are 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And you remember them as ATGC, because A always binds with T with a double bond, and G always binds with C with this triple bond, and this is a complementary base pairing principle. So if we study the structure of DNA, it's a double helical structure. It's a polynucleotide. What is a polynucleotide? A nucleotide is one component, one monomer, which is a phosphate, deoxyribose, and a nitrogenous base. Many nucleotides, they combine by a condensation reaction and form a polynucleotide, and DNA is a polynucleotide. And one base is making the hydrogen bonds with another base on another strand, and adenine is always next to thymine with a double bond, and guanine is always next to cytosine in a triple bond. And you make sure you remember this: what is a nucleotide, what is the structure, and complementary base pairing principle. Okay, now what is a human genome? Now you can see that there are a lot of base pairs in a human DNA, and Human Genome Project, genome is the complete genetic material of the organisms, and there's a Human Genome Project, which is an international collaboration to sequence around 3 billion bases and more than 20,000 genes. It is important to understand the genetic diseases and the inherited disorders. It is also required to produce better personalized medicine. The Human Genome is important. Genome Project is important to understand evolution and identify the new drug targets. So you should know the importance of Human Genome Project and make sure you elaborate these points. Next is protein synthesis. That's for the higher tier. Now, when you have a DNA, the DNA is converted into the mRNA, and this process is known as transcription, and that takes place in the nucleus of the cell. Then this messenger RNA leaves and reaches the ribosomes in the cytoplasm, and these messenger RNA code is read and an amino acid chain is synthesized. So the DNA copies the code to form messenger RNA by the process of transcription. The messenger RNA leaves the nucleus and reaches the ribosome. The ribosome reads the bases in the sequence of 3 bases, and the transfer RNA brings the corresponding amino acid and forms a polypeptide chain. The polypeptide chain then forms a protein. And always remember, the bases are always read in triplets, and this is known as codon. AUG is corresponding to a code of methionine. So the tRNA brings methionine. Then the next is red, which is for phenylalanine. So phenylalanine comes in, and you can see that the polypeptide chain is growing. In this way, a long chain of amino acid is made by reading this triplet, and these amino acids make a polypeptide, and that gets folded to form proteins and enzymes. Okay, so I hope this concept of protein synthesis is clear to you. Now, you must have seen now that DNA forms an RNA, and RNA makes a protein that gives the expression. So what happens if there's a change in the sequence of a DNA? If there's a change in a sequence of DNA, it results in the change in the sequence of mRNA. If the mRNA is changed, the amino acid gets changed. And when the amino acid gets changed, it results in the formation of non-functional protein or enzyme, and that protein will have a different structure and the active site will be different, so the substrate can no longer fit to it and can be non-functional. This is what is mutation. Now, this can be silent because it results to the change of the base that does not make any change to the amino acid. Harmful if it causes any disease, or beneficial, it gives some extra characteristics to an organism. Okay, so you can see in this, our original amino acid chain is all his, but with the change of one amino acid, the his amino acid changed with pro, and that's what caused a malfunctioning protein. So
Okay, so now since we are doing digestion, and digestion is chemical, that is, it involves the use of enzyme, you should know what are enzymes. So make sure you remember this definition and this lock and key model, very, very important. So I'll just take you through it. So enzymes are biological catalysts. So I think in chemistry, you remember what are catalysts? Catalysts are those substances that increases the rate of reaction without being used up. So if there's a catalyst which are used in biological reaction, we call them enzymes. So enzymes are biological catalysts that increases the rate of a biological reaction without being used up. Now, what do enzymes do that increases the rate of reaction? They actually provide an alternative route to a reaction, which works by lowering the activation energy. And enzymes are chemically proteins, so they are very sensitive to heat, pH, and temperature. Okay, now this is the structure of the enzyme, and the enzyme has a specific shape, and it has a site called the active site. Active site is a site where substrate molecules bind to it. Now substrate and the active sites, they have complementary shape. Remember to use the word complementary. Most of the time in the exam, students say substrate and active site have same shape. No, that's wrong. It's like a lock and key. The substrate and the active site, they have a complementary shape. The substrate binds to the active site, forms an enzyme substrate complex. The enzyme substrate complex react to form an enzyme and the product, and it disintegrate to give the product back, and your enzyme gets regenerated. So remember, enzymes are highly specific due to the active shape. As the active shape has a specific shape which is complementary to the substrate, so the specific substrate molecule can fit into the active site of the enzyme. Now, what are the factors that affects the enzyme activity? Temperature and pH are one. We'll be discussing that. But the substrate concentration and enzyme concentration also affects the rate of reaction. So as we increase the substrate concentration, there'll be more molecules to bind with the active site. Therefore, the rate will increase at the start. But after that, it levels off because all the enzyme active sites will be used up, and then enzymes will become a limiting factor. The same thing work with the enzyme concentration. As we increase the enzyme concentration, there'll be more active sites for the substrate molecules to bind to. But after that, further increasing the enzyme concentration will have no effect because then all the substrate molecules will be used up. So substrate will become limiting, so the graph will level off. Now for the temperature, as we increase the temperature, the rate of enzyme catalyzed reaction increases because the particles gain kinetic energy. As per chemistry, they'll collide more the collision, more the chances of reaction. It increases up to a point, and this point is called the optimum. Optimum is the point where the enzyme activity is the highest. But after that, the rate will drop suddenly. Why is that? This is because the enzyme gets denatured. Now, what is denaturation? Now, denaturation is when you heat any specific enzyme due to being protein in nature, their bonds break. So they change the shape. Now, when they change the shape of the active site, so the substrate molecules can no longer bind to it, decreasing the rate of reaction. So denaturation is change in shape of the enzyme with a change in pH or the temperature. The same effect happens with the pH. As you increase the pH, it increases the rate of reaction up to the optimum, and after that it drops. It doesn't drop that suddenly like temperature, so we make like a bell-shaped curve, and then it again drops. Crystal clear to you, and do exam questions on this topic. Okay, so I hope you have understood this video well. If you still have any doubts, leave a comment below. We'll reply you as soon as possible, or you can go to our website where we have 24/7 chat support, where you can post any queries before your exam and you can get instant reply. You can go to my website where you can find the notes of this as well. The link is mentioned in the description box below. If you like this video, then choose subscribe to our channel, and do not forget to like, comment, and share this video. And if there's any specific topic you want me to make a video on, do leave a comment below, and I'll make sure I'll put that up before your exam. Okay, so please go over this topic very, very seriously. It's a very important topic, and I'll see you next in your next video. Till then, happy revising.